Good evening, everybody, and firstly, huge thanks to Charlie for inviting me to come and talk and to share some of this research with you. And thanks to all of you as well for turning out on a cold, slightly wet Thursday evening as well. So I hope you find this useful. After Chloe and I have done the presentation, we are going to hang around a little bit. We're very happy to answer questions about the work. So please feel free to come and ask those questions if you have any. So I'm going to talk about a particular study that started way back in 2018, and just a bit of admin first before I get into the meat of what we actually did. The study was funded by Albarada Trust, and a lot of you in the room probably recognise that name. Research isn't cheap, and the Trust put not far of £500,000 into this project, actually, just to give you an idea of the scale, the cost of this sort of work. Very generously funded, and we're very grateful to them for doing that. And I hope we've repaid that investment by coming up with some really interesting findings, which in the not too distant future are going to lead to some real benefits for foals and foal health. Like all research that happens in universities, we follow very strict ethical and integrity guidelines. And just to reassure everybody in the room that this project was fully reviewed by the university's ethics committee. Chloe and I are just going through that process once again for the follow-on study, and they ask what seem like the most nitpicking of questions, which can be really frustrating, but they do make sure that all the research that we do is being performed to the highest welfare and ethical standards. And I also want to start right up at the top of this presentation by thanking all of the vets, Charlie and his team included, who provided us with absolutely essential contacts within the breeding industry. And huge thanks to Whitsbury Manor and the other stud farms who allowed us to take samples from their foals and who provided health data as well for this study, and really critically gave us feedback on the data as well. They really helped us to interpret the data and to understand what that data was telling us about the health of foals, what that might mean. So that will become clear as the presentation progresses. So a little bit of very general background to start with. Many of you in the room will be aware that the gut microbiome, that is the bacteria, also viruses and fungi to some extent, but particularly bacteria that live in our guts, those have been the subject of quite intense scrutiny, investigation and research over the last 15 years probably. Now there are two very big projects that I want to mention here. I told you that our project cost around about £500,000, which of course is a very big figure. But the MetaHit project, this was a massive European Union consortium project. This was funded to the tune of hundreds of millions of euros. It ran over 5-6 years, can't remember exactly. They're just in their second round of funding now. Huge amount of investment in understanding the gut bacteria of humans, and how it affects so many aspects of our health and livelihoods. Also, in America, this human microbiome project was spending hundreds of millions of dollars of American money, not doing overlapping work, but doing related projects, understanding the gut bacteria of humans, understanding how it affected health, and how it could be manipulated to enhance health. So we're really riding on the coattails of many of the medical researchers who are making astounding findings about the importance of gut bacteria. For us, we're asking the question, how does this affect horses? If gut bacteria are important in humans, they must be equally, if not more important in horses, because the horse has a massive community of bacteria living in its gut. So that's what we set out to investigate. So the study that I'm going to tell you about was called the Well-Foal study. It ran from 2018 to 23. It was the 2018 foal crop that we used to conduct this study. We worked with 5 different stud farms. We wanted to have a mix. We didn't want all our foals to come from the same farm, because we wanted some variation within the population of foals. And we recruited 52 foals. We actually started with a few more than that, but we lost one or two on the way. We wanted to follow them for over 3 years, so that was quite a challenge for us to recruit those foals, study them very early in life, and then to continue to collect health data from those foals as they got older, as they went into training, and as they started racing. So from these foals, we had almost 450 fecal samples. Now, hopefully you can all see it. Let me turn that around just in case it's obscuring the vision of some people. The chart that you can see here on the right of the slide, this represents our sampling programme. So the blue line here represents the first year of the foal's life, and each of these arrows is a sampling point. At each of these times in the foal's life, we took a fecal sample for analysis so that we could understand, so that we could characterise, measure and describe the gut bacterial communities that were there at that point in time. So I won't read out all of the times, but you can see they're particularly clustered very early in life. 2 days old, 7 days, 14 and 28 days old, because we knew from human studies and some work that had happened in horses, we knew that this early life period was really critical. So one of the things that I hope you're going to take away from this evening is understanding that these first few weeks of life for the foal are really critical. The outcomes that we were interested in were health outcomes. We wanted to know how the gut bacteria in these very, very early days of life, how that affected the long-term health of these horses. So we collected health data through our veterinary surgeons. They provided us with monthly health data from all of our 52 foals on the study. And we collected that for the first 3 years of life. So very, very early collection of data about gut bacterial communities, and then longer term collection of health data. We also collected, because we're dealing with race horses, these were all thoroughbreds bred to race, we were also able to collect performance data as well. And that was a different part of the analysis, which I will tell you about also. And we could look at links between what happened very early in life in gut bacteria and the ultimate or the subsequent racing performance of these animals. So in the olden days, when we wanted to know about bacteria, we used to put them onto culture plates and we used to grow them in the laboratory and identify bacteria. And that works pretty well for some things, but it is limited. We know that the bacteria that live in our guts and in the guts of horses, a lot of those won't grow very well outside of the body, outside of the gut environment. So growing them is very difficult, and we know that if we do that, we miss a lot of bacteria. So the technology that we used, which is technology that's only become available in the last 15 to 20 years, is that we use gene sequencing. So from the fecal sample that the foal produces, we extract all of the DNA from that sample. So all of the DNA goes into solution. So that's DNA from the foal cells, from bacteria, from plant material. We're then able to select from that DNA just the DNA that is specific to bacteria. And that bacterial DNA then goes into a very clever machine, which I have absolutely no idea how it works, and it gets sequenced. So the DNA sequence, little fragments of that bacterial DNA is sequenced, and that data comes back to us in a huge file of data. There are usually hundreds of thousands of reads of little bits of bacteria. Using computer programs, computer software programs, we can then compare those little fragments of DNA. We can sometimes stitch them together to make longer bits of DNA. Then we can compare them to a database of known bacteria, and we can say, yep, that fragment is from this particular bacteria, that fragment is from another bacteria, and we've got 200 of this particular fragment, which we know is from that bacteria, so we know the abundance, we know the amount of that bacteria that is present. So it's really powerful technology, and it gives us a really good overview of what's happening in terms of these gut bacterial communities. Now, this is a slightly complicated figure. Don't panic, I'll talk you through it. Once you understand what's happening, it gets a lot easier. So, the data that I'm showing you here is from the 9 different sampling points, the 9 times that we sampled our foals throughout the course of the study. So if you remember, this was 2 days old, sample point 2 was 7 days old, sample point 4, which is an important one, I'm going to come back to it quite a lot, sample point 4 is 1 month old, 28 days old. Now, within each of these blocks, there are 52 columns. Each of those 52 columns represents the bacterial community, the distribution of different bacterial families within the gut contents of one particular foal. And those different bacterial families are coloured. So it doesn't matter about the names here. There are lots of unpronounceable Latin names. Doesn't matter. The fact is that different colours represent different bacterial families. So if we start up here actually at 1 year old, we can see that the distribution of these families is relatively similar across our 52 different foals. If you have a look at the amount of this blue bacteria down here, there's roughly the same amount in all of the foals, and then similarly we've got a green bacteria just above that. And if you then compare that whole block, sample point 9, compare that to the sample point before, the pattern of colours looks roughly the same as well. So what that's telling us is that the bacterial community between this sample point here, 8, which is 9 months old, and 1 year old, it hasn't changed very much. So the gut bacteria is relatively stable at that point. But if we look at the other end of the foal's life, if we look at very early life, what we see here at 2 days old is quite chaotic. Different foals have very, very different patterns of gut bacteria. And if we then have a look at 8 days old, we can see there's a bit of change, but there's still a lot of this red stuff in there, not so much of the blue stuff down here at the bottom. But then again, sample point 3, so this is 2 weeks old, we can see again there's maybe a little bit of organisation happening down here with this blue bacteria, which is starting to approach this sort of level. But if we have a look at sample point 6, by the time we get here to 3 months old, then this is looking relatively stable, and the 3 months old pattern, the distribution of colours here, is roughly the same as 1 year old. So broadly speaking, we can say that this data is telling us that by about 3 months old, the foal's gut bacteria, after a period of instability and change, it's established an adult-like profile. So your foals, when they reach that age, then things are getting pretty stable, really. But before that, there is quite a period of change as the foal starts to develop its first bacterial communities, and those then evolve and mature. Of course, the foal's feed, its nutrition is changing during that period as well. So it's moving from a very milk-based diet in the first days and weeks to a much more fibre-based diet. And that's probably responsible for some of the change that we see, but there are other reasons as well. Okay, I just want to introduce, well, not introduce, but just explain the meaning of this term diversity, which I'm going to use quite a lot in the context of bacterial communities. I'm sure you all understand the concept of diversity, but perhaps haven't heard it used in the context of bacteria. So if we look at mixed bacterial communities, the diversity of those communities is simply the number of unique bacterial species or families that are present within that mix. Some will be present in relatively high abundance, so there might be a lot of one particular bacterial species, there might be a lot less of another. So within that overall diversity, there are differences that can be measured. But generally speaking, in most of the human work and the laboratory animal work that's been published, generally speaking, high levels of bacterial diversity in gut bacterial communities are associated with good health. So diversity is generally a good thing in the context of gut bacterial communities. Okay, so another slightly complicated graph, but again, don't panic, I'll talk you through this. So what we're looking at here is a summary of our data. Now, if we start with this graph over here on the left, this is data which is looking at foals that have been separated according to their gut bacterial diversity at 1 month old, sample point 4. So we've divided the foals at this point into the blue foals that have high levels of gut bacterial diversity, and you can see there are 17 of these foals within our population, and those foals that have the lowest level of gut bacterial diversity, and there are 18 of those. So we've got two different groups of foals: high gut bacterial diversity, low gut bacterial diversity, when they are 1 month old. And what this graph does is that it shows the diagnosis, the occurrence of respiratory disease. So veterinary intervention that are diagnosed as being respiratory disease. So typically that could be where a stud farm manager asks their veterinary surgeon to examine a foal because it's coughing, or it's got a nasal discharge, or maybe it's just a bit off colour, it's a bit dull and depressed, and the veterinary surgeon examines the foal and decides it's likely to be a respiratory problem. So those are recorded on our database. Now, at starting point here, none of the animals have had an episode of respiratory disease. So we've got the age of the foal down here on the bottom axis. So first 50 weeks, roughly the first year of life, up here to about 3 years old. Each time we get an episode of respiratory disease, that is marked by a down tick, a downward notch or a step in the line for these animals. And what I hope you can see here, the blue line represents the foals, the 17 foals that have high levels of gut bacterial diversity. You can see the gradient of that line is relatively shallow. There are not many foals that have an episode of respiratory disease during that 3-year period. That is very different to the red line, where there is a much steeper gradient, which means the rate at which these foals are getting respiratory disease is much greater. And ultimately, a larger number of these animals, these foals with low bacterial gut diversity at 1 month old, a much greater proportion of them, nearly 75%, have had one or more episode of respiratory disease intervention for a respiratory problem by the time they get to 3 months old. Now, something similar is also happening in this graph on the right-hand side. Again, we've divided foals into those with high gut bacterial diversity and low gut bacterial diversity. This time, the data is taken from sample point 8. So if we have a look down here, that's about 9 months old for these foals. And if you have a look at the vertical axis here, we're looking at orthopedic disease this time, rather than respiratory. So orthopedic disease, gut bacteria, doesn't seem like a natural fit. But what we do see is that, again, this blue line, we can see that those animals with high gut bacterial diversity have a relatively low rate of developing orthopedic problems. And this was orthopedic problems of all sorts, including fractures when these animals went into training and when they started racing. But if you compare that to the low gut bacterial diversity, you can see there's a much steeper rate of occurrence of these orthopedic problems. So you can see particularly happening there, particularly around that transition period as their yearlings, they're going into their 2nd year of life. Now, we can do some fancy statistics on these to find out whether this is a real finding or not. I wouldn't begin to understand the mathematics behind it, but just to point out that these are statistically significant findings. So that means that the data, the likelihood is that this is a real finding, it's not a chance finding. Something similar here, I won't labour this one, but we found something similar with soft tissue injury and disease as well. So these are things like abscesses, patent urachus, soft tissue problems that we get in foals and these horses as they get a little bit older as well. And we found that particular bacterial families this time, so we're not looking at diversity here, but we're looking at the abundance of specific families: Streptococcaceae, Pseudomonasaceae, at very, very early time points, we found that horses that had high levels of these particular bacteria had an increased rate of soft tissue injury and disease as they went through their lives up to 3 years old. So we found these two associations, one between gut bacterial diversity and the other types of association between particular bacterial families that are present very early in life that are then associated with an increased risk of disease as the animals get older. Okay, I'm going to move on and talk about the athletic performance findings now as well, because obviously we were dealing with a population of foals bred for racing, the majority of them went off into training and then into racing, and of course that data is readily available for us. But how do you measure athletic performance? Veterinary scientists have tried to do this in a number of different ways, and there is no perfect way of doing it. All ways are flawed. So we actually landed on a strategy of using 3 different measures of performance, all better or worse in some degrees. So we looked at the official BHA rating for each of these horses. We also looked at a relative race placing for every run that a horse had. So 1 being the best, 0 being the worst position in the race. And this is a way of standardising this particular metric for the number of runners that might be in the race. And then another measure, which is perhaps a bit more intuitive, race earnings. Again, it can be taken as a crude measure of performance ability in these horses. So these were our 3 outcome measures. And what we were interested in was whether there was any relationship between these as indicators of performance in these horses and their early life gut bacteria. And of course, I wouldn't be talking to you this evening if there wasn't a relationship there. So there is. Let me explain this to you. So each of these grids contains the outcome, the results of a lot of data crunching. So at each of our different time points there, we asked the question of a computer program. We asked whether there was any significant association, any link. Broadly speaking, you might call it a correlation. That's not quite accurate, but a link between these different measures of gut bacterial diversity. Just as you can measure racing performance in different ways, you can measure gut bacterial diversity in different ways. So we looked at 7 different measures of diversity, and if the computer program saw that there was an association between gut bacterial diversity and the particular outcome variable, in this case we're looking at official rating. If it was a positive association, it returned a red splodge for us. If it was a negative association, it was a blue splodge. But most importantly, if the dot has a black line around it, it means it's statistically significant. So it's very unlikely to be a chance finding. The computer program has a high level of confidence that this is a genuine finding. And if we look at this left-hand pair of graphs to start with, so here we're looking at official rating and the link between gut bacterial diversity and official rating, and you can see that all 7 of our variables are showing up as being positively associated with official rating at sample point 4, so 1 month old. So this is saying gut bacterial diversity at 1 month old is associated with the eventual official rating that these horses receive once they start racing. And just in the graph below, I've presented that perhaps in a slightly more intuitive manner. So on the bottom axis down here, we can see the measure of diversity. So diversity increasing from left to right. And here we have our performance measure, official rating, increasing from bottom to top. And you can see the angle of that line shows you that the nature of this association as gut bacterial diversity at 1 month old, as that increases, so the eventual official rating that these horses gets is that's increasing also. There's then a similar story for our other performance variables. So standardised placing in a race, we can see there are associations with diversity at 1 month old, and also here 3 months old, I think this is. And again, you can see the slope of the line, and a similar story for earnings. Again, this sample point 4, 1 month old, seems to be really important. There's this association. What's happening at 1 month old is somehow influencing the athletic performance of these horses. Okay, so moving on to something, a slightly different aspect of the study now. Some of the foals on our study received antibiotics during the early stages of their life, the first few weeks and months of life. And we were interested to look at how this might affect the gut bacteria. We know that when we give antibiotics to horses, or to any animals, to humans, we know that it does influence gut bacteria. It kills a lot of the bacteria, and the likelihood is that it depresses bacterial diversity. So looking at the data we had from our study population, we divided the horses into 2 groups. Those that had received antibiotics during the first month of life and those that hadn't. And when we then looked at some variables, there's some interesting patterns that emerged. So looking first of all at gut bacterial diversity in these animals. So this is diversity at 1 month old. Those that had received antibiotics had a significantly lower level of gut bacterial diversity. If you remember from the previous slides, that's not a good thing. If we look at official rating as a measure of performance, we can see there's maybe a slightly lower level of official rating achieved by those horses that received antibiotics early in life, but it's not statistically significant. But what is significant is that earnings are significantly lower in those animals that received antibiotics during the first month of life. So there's certainly a suggestion there that giving antibiotics early in life, suppressing gut bacterial diversity, that might have a knock-on effect on the racing performance of those animals when they eventually reach the race course. We also looked at health outcomes. So again, coming back to respiratory events, which we were particularly interested in. These are animals that had antibiotics given to them in the first month of life. We can see those that did have antibiotics had a higher rate of respiratory events, so they were more prone to respiratory disease than the blue animals that didn't have antibiotics during the first month of life. So some interesting insights there, just from a relatively small subset of our study, into how giving antibiotics early in life could potentially impact both health outcomes and athletic performance outcomes. So I've shown you a lot of quite complicated data, but I think I've just got 3 slides with very, very clear conclusions that we can make from this study. The first is that high gut bacterial diversity, particularly around 1 month old, is associated with both a reduced risk of disease and better racing performance up to 3 years old. We can't say anything about after 3 years old because we didn't study that. That wasn't part of our study. We've also identified particular bacterial species which, when present in the gut bacteria of very young foals in the first few weeks and months of life, are associated with particular health outcomes. So either an increased or a decreased risk of particular types of disease. So this could be really useful information. I'll come back to this a little bit later in the presentation. And then the third conclusion is about the antibiotic treatment. So if we do use antimicrobials in the first month of life, we know that there are some potentially undesirable impacts associated with that, particularly around risk of disease and potentially reduced performance potential in those animals as well. Okay, so those are the conclusions from the study. That's, if you like, the first bit of what I'm going to talk about. As with any bit of science, this raises lots and lots of questions and doesn't necessarily provide us with a lot of answers. So working with Albarada Trust, we've actually got a second round of funding to continue this work and to address some of the practical questions that arise from the work that we've done. I'm going to start off telling you about some of these, then I'm going to hand over to Chloe for another part of the project that is just starting at the moment, just underway. So some of you might not recognise it, but on the left-hand side there, that's the gut of the horse. It's a laboratory model of the horse. And we've got 3 chambers here, 3 fermentation vessels, which represent 3 parts of the horse's hindgut. The cecum, and then 2 different parts of the horse's colon. This is the bit of the gut we're most interested in because it really does the majority of the work in the horse in terms of nutrient digestion, in terms of production of metabolites, and it's where most of the bacteria actually live. This laboratory model pumps in this brown sludge, which is broadly represents feed material. There's a pump here, it goes into the top chamber, then it sits in there for about 48 hours. There's a steady trickle of nutrient material into the vessel, and then material out of this vessel into the next vessel. And this represents the normal throughput of ingesta through a horse's gut. We control the temperature of these vessels because we know that it's going to be at 37, 38 degrees, the horse's normal body temperature, so we want bacteria that are attuned to growing at that temperature. And these controllers down here, they maintain the pH, the acidity of these 3 different vessels as well. And we know that in the normal horse, the pH of each of these parts of the horse's gut is slightly different, which will suit some bacteria and not other bacteria. So by maintaining those different conditions, by maintaining temperature and different pH, we can selectively grow the bacteria that will grow in different parts of the horse's hindgut. So this doesn't produce exactly what happens in the horse's gut, but it produces a pretty good imitation of it. And it's certainly a useful thing to do in the laboratory for all of the reasons that I've listed here. It's a way that we can recreate what's happening in the horse's gut in a cheap, convenient manner, and one that we can manipulate very easily. So in this second part of the study, what we're doing is using this benchtop model of the horse's gut to investigate how antibiotics affect bacterial communities. So we'll be able to get much more of a handle on this antibiotic issue that we noticed. We will also be able to see how we can mitigate against the damage caused by antibiotics. So as well as adding nutrients to these vessels, what we will do is add antibiotic to mimic veterinary treatment with antibiotics, and then we'll see whether there are perhaps probiotics that we can add to the mix that might limit the harm or minimise the harm that's being done by the antibiotics. And there might be other feed supplements that we can add here as well to investigate their ability to mitigate this antibiotic-mediated impact. Different antibiotics will have different effects. So something that veterinary surgeons would be really keen to know is that if you have to use an antibiotic in a 2-week-old foal, which is the antibiotic that causes the least damage to the hindgut bacteria? That's the sort of question that we'll be able to answer with this gut model. Over to you, Chloe.
We know that in the first few months of life are extremely important for our foals and their gut microbiome. But what we want to know now is how do they develop this gut microbiome from such a young age? And obviously we've looked at the foal, but we haven't looked at the mare yet. So essentially, this part of the study, we're going to be looking at routes of transmission from the mare to the foal after birth, essentially. So the aim for this study is that we're going to sample mares 2 months before they give birth. So essentially, we're going to look at the microbiome of the mare and see how 2 months before birth and 2 months after birth, the gut microbiome can change during pregnancy. And we're also going to be looking at different ways that the mare can potentially transfer microbiota to the foal. So the things that we will be looking at include feces, because that's a very good representation of the gut microbiome, which is what we use for the first study. and we'll be looking at saliva, because when the mare obviously gives birth to the foal, she licks the foal, spreading all the microbiome from her mouth onto the foal. And we're going to be looking at the microbiome around the mare's teats, because this will be one of the first things that the foal latches onto. And we think potentially that the bacteria around the mare's teat will seed into the foal's mouth, essentially, hence why we will be looking at also the foal's saliva. And we will also be looking at milk, because in humans it's been shown that milk has a range of benefits, such as providing antibodies and also bacteria, and it helps just kind of set the baby or foal up for life. So that's essentially what we'll be doing. So it's my role to be collaborating with all of our lovely vets and studs and organising the sample collections. So we will be collecting fecal, saliva, udder skin and milk samples, which will be then taken back to the lab to do similar to what was done in the first part of the study. So DNA extractions from each sample type, which will then get sent off for complex sequencing, so we can analyse the microbiota of both the mare and the foal, and potentially how the mare has impacted the foal's microbiome, essentially. So that's this aspect of the study. Anything else to add, Chris?
No, that's great. Thank you, Chloe.
Thank you.
So that's going to be your life for the next 6 months,
Yes.
isn't it? Collecting all of these samples. Yeah. But there's some really exciting possibilities that might come out of this. So, for example, we might be able to improve the gut health of the foal by devising a cream, a probiotic cream that is put on the udder of the mare. That's the sort of thing that we're thinking about here, the practical implications. A further way in which we are exploring practical implications or practical solutions to this issue of foal intestinal health and enhancing intestinal health is with this particular project, which is funded by the Horse Race Betting Levy Board. This project is it's a 3-year project. We're 1 year into that project just now. So I'm sure many of you have used probiotic products in your foals and possibly your mares as well. Most of the probiotics, in fact all of the probiotics that are available, that are sold for administration to horses now, are repurposed probiotics, mainly from the human market and occasionally from the animal market. None of them have been developed specifically for horses. Now, particularly with human probiotics, the human intestinal tract is very, very different from the horse's intestinal tract. Why should a probiotic that works in humans work in horses? Some of them do, and we understand the reasons behind those, but we don't have any probiotic products that are engineered specifically to benefit horses. So we now have the data from the foals that I've just described to you to identify which bacterial species are associated with good health outcomes and also good racing outcomes in young foals. So what we're doing now in the laboratory is trying to culture those bacteria, so trying to grow them in the laboratory, and we can then subject them to various types of testing to see whether they might be potential candidates to be developed as probiotics. So we're looking at the genes of those bacteria, and that can tell us a lot about the metabolic products from that bacteria, and also some of the bad things, like do they carry antibiotic resistance genes? If they do, then we don't want to be feeding those as probiotics. So that's a useful screen. And we can also look at the phenotype, which just means really the metabolism of the bacteria. Is it a bacteria that's going to survive journey through the stomach? Is it resistant to bile in the intestine? Some of the other metabolic things that we know differentiate good probiotic bacteria from those that really aren't going to work. So at the end of the day, at the end of the screening process, we hope to have a list of candidate probiotic bacteria that can be further developed for administration, particularly to foals. We've got a short list of about 138 bacteria that we're screening at the moment. And as we go through this screen, so that short list will become smaller and smaller as various bacteria are ruled out. But included in one of the stakeholders, or two of the stakeholders in this project, are probiotic manufacturing companies as well. So right from the start of this project, right from the design of the project, we've been talking to them about their requirements. So what we're doing here should be fairly readily convertible into probiotic products when this project comes to an end.